This method can help answer key questions in the chromatin architecture field, such as how the 3D structure of chromatin can influence gene expression and development. The main advantage of this technique is that it provides a high-resolution view of chromatin architecture in precisely staged fly embryos.
Though this method can provide insight into chromatin organization in development, one can also use existing lines from transgenic fly libraries to test their effect in chromatin organization.
To begin the protocol, adjust the total volume of previously collected embryos to 2 milliliters with PBST. Add 6 milliliters of heptane and 100 microliters of 37% formaldehyde in water. After adding formaldehyde, vigorously shake the tube up and down for 1 minute. The aqueous and organic phase will combine to form a shampoo-like consistency. Agitate the mixture on a rotary mixer for 10 minutes. Centrifuge the tube at 500 times G for 1 minute at room temperature to collect embryos at the bottom of the tube. Aspirate the entire shampoo-like liquid and discard it, taking care not to aspirate any embryos. 15 minutes after the addition of formaldehyde, resuspend the embryos in 5 milliliters of PBST with 125 millimolar glycine. Shake the embryos up and down vigorously for 1 minute. After centrifuging, aspirate the supernatant. Using a 1000 microliter pipette, transfer a batch of 100 embryos to a small glass vessel suitable for sorting, preferably on a dark background.